Okay, my favorite subject, mitochondrial disorders. Just kidding. Anyway, I'm just going to be discussing the genetic components of mitochondrial disorders, which usually give me a headache. But anyway, so mitochondria are intercellular organelles, and unlike a, a lot of other organelles that we have in the cytoplasm, mitochondria have their own DNA. It's actually hypothesized that there's a symbiotic relationship between the mitochondria and the cell, and they were once a single cell organism that ended up inside of our cells, and we work together. The mitochondria make energy, and we help make the mitochondria. So in addition to their own DNA, there are over 200 nuclear genes in our genetic components that help create the mitochondria. So let's first talk about the mitochondrial DNA. So again, the genetics of mitochondria are complicated because there's mitochondrial genes and there's nuclear genes that have to do with the mitochondria. So there are 37 genes in this circular DNA. 13, they code for 13 proteins, 22 tRNAs, and 2 rRNAs. 13 mitochondrial gene-encoded proteins all instruct to produce enzymes in the oxidative phosphorylation system. So the mitochondrial genes are primarily involved in the enzymes of the oxidative phosphorylation system. And this is the system involved in creating energy. Common mutations include MELAS, MERF, LHON. So organs affected by mitochondrial mutations. Organs with high energy demands are the most affected by abnormal mitochondria. And this includes the muscle, the heart, which is the most important muscle in our body, the brain, and senses such as vision and hearing. When a patient has a variety of seemingly unrelated problems, mitochondria should come to mind. And again, you can see skeletal muscle weakness, heart problems, not just the muscle, but also the electrical system, the eyes, eye movements, as well as retinopathy, liver, kidney, Fanconi syndrome, pancreas, diabetes, Pearson syndrome in the blood, sensorineural hearing loss, pseudo-obstruction of the GI system, and seizures, myoclonus, ataxia, stroke, dementia, migraines. Again, all these seemingly unrelated things can all be caused by the mitochondria. So let's talk about those 37 genes in the mitochondria. Those are inherited exclusively from the oocytes. So the oocyte has all the cytoplasm, therefore all the mitochondria. And the sperm just donates DNA. So when a mother is affected with a mitochondrial disorder, 100% of her children will be affected. If a son is affected, 0% of his children will be affected, but 100% of her children will be affected. So these two individuals are affected. The son does not donate his mitochondria to either his daughter or his son, who do not donate those mitochondria to their children. But just to make things even more complicated, I apologize, is sometimes when I showed you in that last slide, that's a mother affected with a mitochondrial disorder. But sometimes, again, there are hundreds, maybe thousands of mitochondria in the cells. And so sometimes there are some in the oocyte that are not functional and some that are. And there's something known as a bottleneck effect. Again, when the mother either has mild disease or no disease, she could put all of her bad mitochondria into the oocyte, like maybe 80% of them are abnormal, and they're going to have severe disease. She could have a little bit more than she has, or the child could have a very small percentage of abnormal mitochondria and not have symptoms. So you could see a family with a variety of symptoms based on how many abnormal mitochondria end up just by bad chance into that offspring. But once a female has abnormal mitochondria, has that threshold, then she's going to pass it on to her offspring. So again, here's a little bit more complicated of a pedigree that this mom has no symptoms, but a couple mitochondria that don't work. And these two children got, the vast majority of abnormal mitochondria whereas these two children did not, but she, unfortunately, passed on more abnormal mitochondria to her offspring. So that, again, is the bottleneck effect, and that makes this pedigree a little bit more complicated than the first one that I showed you. That's pretty straightforward. So I'm going to leave to biochemistry to go over all the nitty-gritty nuts and bolts of oxidative phosphorylation, because they're far better at that than me. But about 20% of proteins responsible for oxidative phosphorylation are encoded by the mitochondrial genome, and point mutations can result in non-functional proteins that affect this system. So let's talk about some of the more common mitochondrial disorders that you need to know, and one is mitochondrial encephalomyopathy, lactic acid and stroke, or MELAS. That's why I bolded all the first letters for these. This is the most common mitochondrial disorder, occurring about 1 in 4,000 individuals. It's caused by a mutation of the mRNA in the mitochondrial genome. Symptoms usually begin in childhood, and they include muscle weakness, hemiparesis, vision, seizures, migraines. Again, lots of systems that have to do with what mitochondria do. Lactic acid is often elevated, and that's usually because of the muscle issues. And if they do a muscle biopsy, they see something called ragged red fibers, which can also be seen in MRF. Febrile illness can be triggered, it can trigger these symptoms. So maybe they're kind of doing okay when they're healthy, and then they get sick and things are worse. And then we have MRF, myoclonic epilepsy with ragged red fibers. Again, ragged red fibers can be, it's a pathological sign of a mitochondrial disorder, and this is a mutation of the mitochondrial TK gene. Symptoms usually begin during childhood and includes muscle weakness, ataxia, seizures, optic atrophy, and hearing loss. So sounds rather similar to MRF, it's just a different gene. Leber optic neuropathy. So these first two that I was talking about, often this is a pretty severe disorder that individuals with severe disorders often don't reproduce. Although, again, with the bottleneck, the mother could have some kids with more severe or milder phenotype. So LHON, Leber optic neuropathy, is characterized by rapid onset of blindness in early adulthood, and it's caused by the mitochondrial gene ND4. And there's also kind of a sex bias to developing the blindness. 10% of women with this abnormal gene will develop blindness, whereas 50% of males. So not everybody, will develop blindness with this. But again, when you see that a mother, most of her children have, either blindness or symptoms of it, whereas none of the fathers will, because this is an actual mitochondrial disease. So next I'm going to move on to mitochondrial disorders of Mendelian inheritance. Like I said, just to make life a little bit more complicated, there's over 200 genes that are involved in the structure and function of the mitochondria that come from our nucleus. And nuclear mitochondrial disorders include something called Kearns-Sayre, Leber optic neuropathy, Lace disease, pyruvate carboxylase, and pyruvate dehydrogenase, which we reviewed previously. But pyruvate dehydrogenase is on the X chromosome. It's a nuclear gene on the X chromosome, and it's responsible for converting pyruvate to acetyl-CoA. And when the enzyme doesn't work correctly, there's excess pyruvate that ends up getting turned to lactate, which results in elevated lactic acid. Clinical presentations, again, primarily occurring in males, include progressive cognitive delays, low muscle tone or hypotonia, seizures, and abnormal eye movements. And the management includes vitamins that help the mitochondria work better, including B1 and carnitine. As I said, carnitine is a vitamin that's important in both fatty acid oxidation disorders, if you remember from last year, as well as mitochondrial function in general in the citric acid cycle. So it usually is deficient when it's not being used appropriately or efficiently, I should say. And so carnitine supplementation can help. And then pyruvate carboxylase is autosomal recessive. It occurs in males and females. And when you have a child, both parents are often carriers. And again, defer to biochemistry as far as all the pathways, but this disorder presents in the first months of life, and it's a defect in converting pyruvate to oxaloacetate. Clinical features include failure to thrive, and that's why it's in this case, developmental delay, recurrent seizures, and metabolic acidosis. So, clinical approach when considering a mitochondrial disorder is there a number of high energy, are there a number of high energy systems involved? The heart, the brain, the hearing, vision. Does it make sense to anything else? Is there an acute event, or was a virus precipitating this? Does the patient have acidosis? And is there a family history of similar disorders, and what is the pattern? My thought is I have absolutely no idea what's going on, mitochondria has to come to mind and consider testing for. So mitochondrial disorders occur in both the mitochondrial genome and nuclear genomes. Common disorders you need to know is Melos and Murph, and there are over 200 nuclear genes that are also involved. So that concludes this brief.